So my idea is to give you a few examples of the projects that we have in our group, or we had in our group on different aspects of application of systems biology and cancer research. And the first such example is on the topics which can be called formalizing knowledge about mechanisms of cancer progression, about molecular mechanisms of cancer progression. So I'm going to talk about how we in our group or in other groups, how people represent the knowledge about molecular biology in such a way that it can be analyzed by computer algorithms or can be used in mathematical modeling. So these biological networks are represented in the form of more or less complex graphs, which might be of different types. And so roughly speaking, we can say that biological networks is representation of cellular biochemistry at various levels of granularity. One of the examples of such a graph or such a representation is this RECON database, which is close to complete reconstruction of human metabolic network. So currently, this reconstruction contains more than 10,000 biochemical reactions, and each biochemical reaction connects a set of incoming metabolites with a set of outgoing metabolites. So it converts a set of metabolites into a set of metabolites. And all these reactions are connected together. So those metabolites which entered in some reaction and exited from a reaction can enter into the next reactions, and in such these reactions form chains, and all this makes this huge and complex graph, which is called metabolic network reconstruction. So it's important to understand that this is a result of a very careful and long-term reading of many scientific publications in the field, and using this, extracting information from these publications to insert several reactions and put several reactions onto this canvas. But this is only, these metabolic reactions is of course only a part of the story, which can be called cellular metabolism. So let's say on top or upstream of this cellular metabolism, which can include the DNA metabolism, meaning cell proliferation as well, you have this set of signaling networks which connect the interior of a cell with external environment. Usually we call these types of networks signaling networks. And let's say downstream, we have those networks which connect against internal cellular metabolism with genetic program which is encoded into the genome. And there are specific types of networks which can be called transcriptional networks, whose purpose is to read out these instructions and modify correspondingly the properties of intercellular networks, intercellular mechanisms. So in principle, if we have complete reconstruction of such networks, and I would say that they're quite closely, completely reconstructed. Of course, we don't know much still about these networks, but just imagine that we were able, like we reconstructed these metabolic networks, we can reconstruct the rest of the networks, and we have one huge network representing molecular mechanism of a living cell. Then in principle, we have this chemical kinetics formalism, which allows to convert this description into a set of equations, which potentially can be solved, or they can be used to simulate different perturbations applied to a system. So in principle, we have it seems that we have everything to construct a model of a living cell or an organism, but unfortunately we have a huge problem in the definition of quantitative parameters of these equations. So we know something, let's say, about the structure of these networks. We can convert them into equations, but we have very little idea how to parameterize these equations, how to assign quantitative values to the parameters of these networks. And we are very far from completing this knowledge, and actually it's for the moment it looks more or less hopeless. This is why when we say about more or less large-scale modeling of such networks, we talk about some simplified approaches. For example, if we consider only existence of these networks in steady states, so in some states where you have constant fluxes of metabolites through these cellular networks, but let's say the level of metabolites in the cells, they do not change. So this situation can be analyzed using only stoichiometric matrix of the description of these reaction networks, which gives rise to this flux balance analysis. So this is one of the working tool which allows to do large-scale simulations of cellular metabolism. Otherwise, you rely on abstraction and simplification of these networks. So you decompose them into modules, and you describe more or less abstracted interactions between the modules composing the biological system. So there are several levels of simplifications that you can make to represent these networks and make them amenable for different types of analysis. One of the most drastic simplifications of these networks is just take, for example, two proteins or two biological networks and describe the fact if these two proteins interact or not. So this can be described as a graph in which each node represents a protein and each edge represents the fact of physical interactions between proteins. So these types of networks are called interaction networks, and they are the most abundant type of network which is used today in systems biology, including systems biology of cancer. You can make, of course, one step further from this description and introduce into the picture the notion of causality. So you can describe by directed edge of the graph the fact that the change in the state of one protein will cause the change of the state in another protein or in the function of another protein. As such, you can construct these graphs where nodes are proteins and edges are these causal relations, which might be also split into activation and inhibitory relations, and such. You can talk about influence networks, and they're quite abundant in the systems biology field as well. So 20 years ago, there was this dream that we will be able quite soon, I mean, by today, reconstruct at more or less full complexity of the biological network underlying the process of cancer formation. And it was kind of drafted at hugely oversimplified in hugely oversimplified way in this publication that I have mentioned already by Hanahan and Weinberg in 2001. So this is an illustration of from their paper in which they stated that all these 10 hallmarks of cancer, they represent, they are not independent mechanisms in the cell, but they are all interconnected through this more or less complex biological network. But of course, the actual structure of the biological network is way more complex than this simplified description. And many people in the field, they started to systematically collect information about the realistic complexity of this biological network underlying cancer progression. One of the first publications in this direction was in 2005, which described the downstream networks of EGFR signaling in cancer normal cells. Then we contributed with a description of cell cycle in more or less realistic complexity in 2008. Then with participation in our group also, there was this reconstruction of MAPK signaling, which is another fragment of this global network. And then at some point, we collected enough of such reconstructions that we decided to reconstruct, to make a draft of the same picture, but at realistic complexity, which would combine many different reconstructed networks together. So we did the first such an attempt 5 years ago. So we collected all these networks, and actually we drew this representation of this network, some layout on a 2D map. And we provided some mechanisms to browse the content of this map in the way which is very similar to Google, to how we use Google Maps. In reality, we use the Google Map approach to browse the content of these networks. So this resource that we have constructed is called Atlas of Cancer Signaling Network, and it's constantly updated. And I'm going to say a few words about how it was built and what kind of ideas we used to build it. So the first problems that you meet when you try to reconstruct biological networks from reading literature, you have to choose a language to fix this knowledge in some kind of systematic way. Unfortunately, when we read biological publications, we have these kind of small pieces of art describing different fragments of these biological networks in the form of these diagrams. But in reality, you have to decipher these diagrams in order to understand what they represent. So in let's say, engineering field, in electric engineering field, they have this common language for describing electrical circuits. And one of the big advancement of the systems biology field, something like 10 years ago, was introduction of this systems biology graphical notation language, which is similar to this language of electric circuits. So the idea is to introduce a common set of symbols that everybody would understand, using which we can transform these diagrams into something that can be read by anybody immediately. And this was done. So there is this set of symbols which represent proteins or proteins in some modified forms. For example, proteins phosphorylated at specific residues, chemical reactions of different types, and so on and so on. So if you're interested in these topics, I invite you to read the corresponding paper. So just let me give you an example how this works in practice. So this is an example of a reconstruction of a cell cycle mechanism. So actually this is the smallest map of our Atlas of Cancer Signaling Network. And when you open this page from the Navicell website, it's really like looks like a Google map of a country. So what you can do, you can first of all, you can search your favorite protein in the list of protein on the right hand side. And just as you select your favorite restaurant, for example, on Google Maps of Paris, you can select your protein and see where it's located on the map. And then what you can do, you can click on the marker and see some description of this protein, but you can also start to zoom in. And the more you zoom in, the more details you start to see. And in the end, you see quite detailed representation of the molecular reactions where this protein participates. So for example, this set of reactions represent a phosphorylation of ATM protein and then formation of a complex of phosphorylated form of ATM protein with another protein also phosphorylated at a particular residue. Then this map, this complex map, is decomposed into a set of modules, so simplified representations of some fragments of this map, which you can also browse. You can formulate some queries, so you can search for certain words in the description of the proteins. For example, you can type melanoma here and see all the proteins in the description of which you have melanoma word. Or you can type apoptosis, and here it shows all proteins which are potentially involved in apoptosis, because in their description there is a word apoptosis. So, as you know, I mean, Google Maps work using this concept of semantic zooming, such that when you zoom out and you see a kind of map on a larger scale, some details are hidden. And at each level of zoom, you always see only readable information, which is important to represent at a given level of magnification of the map. So we use similar principle to construct, to represent the details of molecular biochemistry. And so as I explained to you, I mean, using when you browse this map, the more you zoom in, the more detailed information you you get. So I stop here, and in the next lecture I will give you an example how biological networks can be used together with molecular data to obtain to improve the analysis of molecular information.